In Mendel's garden, Mendel discovered the principles which founded the science of genetics. He described dominant and recessive traits, but he certainly did not discover every form of inheritance that can be observed in every organism on the planet. And so there are many what are often referred to as exceptions to Mendelian inheritance or extensions to Mendelian inheritance. This certainly does not mean that Mendel was wrong. It simply means that the types of inheritance which can be found in the diverse organisms on the planet, are diverse, and that there was more than what he had originally observed. So in a Mendelian cross, if one individual is homozygous for one allele, say we'll call it the recessive allele, and another individual is homozygous for another allele, let's call it the dominant allele, then we would expect that while the genotype of the F1 generation is heterozygous, where they receive one allele from each parent, that the phenotype would be like that of the parent with the dominant allele. So, we don't at the outset know which is dominant. It could be the blue or the yellow, but we would then expect the offspring to be either blue or yellow, depending on one allele being dominant. That was one of Mendel's three principles or laws, the principle of dominance, where in a heterozygote, one allele would be expressed and one would not. But there are cases where the F1 heterozygotes are neither, say, blue nor yellow, but in between. So they are intermediate between the two homozygous conditions. And so therefore, this type of inheritance is known as incomplete dominance, where heterozygotes have an intermediate phenotype between the two homozygous conditions. So if you crossed a red flower with a white flower and got pink flowers, then that would be an example of incomplete dominance. If two heterozygotes crossed, so here we have two heterozygotes, we know that while all individuals may have both alleles in their cheek cells, their muscle cells, their bone cells, when they make sex cells, this man's sperm will either carry the dominant allele or the recessive allele. This woman's ova will either carry the dominant allele or the recessive allele. And so then there will be four possible combinations in the offspring. A quarter will be homozygous dominant and have one, or homozygous for the blue allele and have one phenotype. A quarter will be homozygous for the yellow allele and have a second phenotype. But then two will once again be heterozygotes and have that intermediate phenotype. In a Mendelian cross with simple dominant and recessives, the phenotype ratio is 3:1, and the genotype ratio is 1:2:1. So in a Mendelian cross, a quarter are homozygous dominant, half are heterozygotes, a quarter are homozygous recessive. So there's a 1:2:1 genotype ratio. But there's a 3:1 phenotype ratio because the heterozygotes look like one of the homozygous conditions. In an incomplete dominant trait, the genotype ratio and the phenotype ratio are the same. And so not only is the genotype ratio a quarter homozygous for the blue allele, half heterozygotes, a quarter homozygous for the yellow allele, but also a 1:2:1 ratio, with half of the population, the heterozygotes, being intermediate between these two crosses. So that is what is known as incomplete dominance. There are a number of examples that could be shown in humans. There's very simple, like the red and the white flower example, often used in biology textbooks. Perhaps the most significant one is that case of sickle cell anemia. So hemoglobin beta hemoglobin is encoded by a gene on chromosome 11. And there is the normal form of beta hemoglobin, but then there is also then an alternate allele, the sickle allele. And we can talk about the molecular aspects of that coming up. And so if you were, say, homozygous for the normal allele, your red blood cells would look like this. If you were homozygous for the sickle allele, your red blood cells could then sickle because the mutant protein would have an abnormal shape, and this would cause the cells to have an abnormal shape. The problem with this shape is that the cells could then get stuck in narrow capillaries and thus block blood flow. And so, blocking blood flow here would cause a stroke. Blocking blood flow here could cause a heart attack. And so individuals with the sickle allele would then have problems with circulation, and this could manifest itself even in children. And so there are two homozygous conditions which we could consider. The homozygous for the normal allele, where blood flows normally because the red blood cells are flexible, or if one substitutes an amino acid, glutamic acid for valine, then the structure of the beta hemoglobin would be changed. This would then change the structure of the cells, making them less flexible, and then giving them that sickle shape. This would then potentially cause them not to pass normally through capillaries, and then potentially to become lodged. The reason that sickle cell anemia is included in discussions of incomplete dominance is if one were to be heterozygous, having one normal allele and one mutant allele, would you be completely normal and free of sickle cell anemia? No. Would you have the disease sickle cell anemia? No. You would be intermediate and have something known as the sickle cell trait. So very often you wouldn't have to worry about any ill effects of sickle cell anemia. But if one were to do very strenuous activities, so for example, when individuals join the military, they are screened to see if they have the sickle cell trait. They might not know. But if they were to go on, long distance runs or start jumping out of a plane and parachuting exercises, for example, then their blood, which had red blood cells with the normal shape, could then start to sickle, and then you could have healthy young individuals now have a stroke because they have the sickle cell trait. And so therefore, this is an example of incomplete dominance. So if two heterozygotes had children, a quarter of their children on average then would have be homozygous for, say, the normal hemoglobin allele. Half would then be heterozygotes. They would have the sickle cell trait, and then a quarter would have two of the sickle alleles, causing sickle cell anemia. And so just as there would be a 1 to 2 to 1 genotype ratio of homozygous individuals for one allele, heterozygotes, homozygous for the other allele, there would also be a 1 to 2 to 1 phenotype ratio of normal individuals, sickle cell trait, where they would be normal most of the time, but could say, if they were going up in altitude, et cetera, experience some of the effects of sickle cell anemia, and then a quarter with sickle cell anemia. Now, before going into other examples of incomplete dominance, it should be noted that incomplete dominance is often a little more complex. And so some of the examples that I'm going to discuss will hit on that. So I'm going to return to this sickle cell anemia example when I talk about overdominance. That is another type of inheritance. And so I know, for a beginning student, this might be a little more than you were looking for. You want, say, an example of incomplete dominance. But so, for example, in this next case is the LDL receptor mutations, which affect your risk of heart disease. Is that an example of dominant and recessive, or is that an example of incomplete dominance? Well, very often the answer, quite honestly, is it could be both, depending on how you look at it. So if you have a mutation for the LDL receptor, that could increase your risk of heart disease. And so we would then refer to that mutation as being dominant, because if you ask which of the offspring would have an elevated risk of heart disease, those who receive 2 copies of the mutant allele would have an elevated risk. The heterozygotes would have an elevated risk. It would only be those individuals which have 2 of this allele which would have a normal risk. So in that sense, this is a dominant mutation. So only 1 mutant allele is then needed. And then it could be, we could say, all right, so in that regard, this is a dominant mutation. But if we looked at it in a different way, you could look at a disease from different angles. You could look at phenotype, you could look at serum levels of a certain protein, et cetera. And so if you ask, all right, well, let's look at cholesterol levels in the blood, you would see that if you're homozygous for the mutant allele, you have a serum cholesterol over 450 milligrams per deciliter. Quite often these are averages. If you're a heterozygote, it could range, say, between 200 and 250, whereas if you're homozygous for the normal allele, between 75, and 175. And so if your classes are normal heart disease risk, elevated heart disease risk, these would all be in one category. But if you're looking at serum cholesterol, you would see that there would be 3 categories, with heterozygotes being intermediate between the 2 homozygous conditions. Also, we could return to this elevated heart disease risk and ask, all right, so even if they all have an elevated risk of heart disease, is there any that have a severely elevated risk of heart disease? So that would be these individuals. And so this is incompletely dominant when one considers the serum cholesterol levels, or it could be incompletely dominant if you split elevated heart disease risk into greatly elevated versus moderately elevated. So sometimes as we classify something as dominant or incompletely dominant, it then varies on how we look at it. I'll get to another human example in a second. While there are some flies and some members of the genus Drosophila where the wild type color is ebony or dark pigmentation, in the species which is commonly used in genetics, then the wild type pigment is more of a tan color. But there are some mutations which will darken the cuticle, like the mutant ebony, the mutant black. These are different genes. And if one were to then perform crosses, one would see that while being homozygous for the wild type allele would give a fly a tan body color, being wild type for the ebony allele would give a fly an ebony body color. The heterozygotes, they're intermediate. They are not as light as this wild type fly. So here's the wild type fly. But they're not as dark as the ebony flies, but would rather be intermediate, especially, so here you see ebony, here you see wild type. But if you were to look at the heterozygotes, especially on the thorax region, then you would see that they do have some darker pigmentation, not as much as ebony. Now, these are sepia ebony flies, so some of them also have dark eyes, but that's not relevant here. So here you can see an intermediate fly. It is not as ebony as ebony is, but especially in the thorax, there are some regions of darker pigmentation. And so this would be an example of incomplete dominance, where the heterozygotes are partway between the 2 homozygous conditions. So once again, this is not ebony, but this is more dark pigment here and there, than one would see in a fly, which was homozygous for the wild type alleles. And so we have homozygous for one allele gives us this phenotype, homozygous for another allele gives us this phenotype, and then the heterozygotes are intermediate with more pigmentation than the wild type flies have. Now, one of the problems in talking about incomplete dominance is it's actually far more common than we think it is. It's just that we call many alleles dominant that aren't. Many of the mutations which we say are dominant are actually incompletely dominant. So before going through human examples, this is actually pretty common. If you were to look at chromosome 2 mutants in flies like curly and plum, on one hand, you could say, oh, this fits the dominant form of inheritance because look, the if a fly receives only one curly allele, it has curly wings. If a fly receives only one plum allele, it has plum eyes. And so having one mutation is enough for the mutant phenotype. That's what we expect in dominant mutations. Also, this isn't skipping generations. Every fly that has, curly wings has a parent who has curly wings. So you'd like to say, oh, all right, so this is dominant, right? Well, if you want to look at it that way, sure. But if you then ask, well, what happens if you have a fly embryo that receives 2 curly mutations? And the answer is it dies. It doesn't make it to be born alive. And so when you're counting flies, you're actually looking at a pool which isn't representative of all of the embryos which were conceived. And so this fly is not normal, but it's not dead either. And so therefore, the heterozygotes with only one curly allele are intermediate between having normal wings and being dead. So even though it would be common to call curly a dominant mutation or plum a dominant mutation, and there are others, and these are important for mapping, as we'll get into later, like dikeet, which is the wings being outstretched, or stubble bristles. These are actually incompletely dominant. It's just that we normally don't get to see the homozygous conditions because they've died. And actually, there's a lot of human examples here as well. And once again, I am going to move to the next type of inheritance in a minute or two. But it's complicated, and incomplete dominance is often in the middle of this, being complicated. So, for example, if you were talking about a gene which can contribute to breast cancer risk, BRCA1, what type of inheritance does it follow? Well, actually, you might see it's actually kind of complicated. So from this point of view, you could see it could be functionally recessive, but then if with loss of heterozygosity, it could become functionally dominant. It could be, inherited this way, so we could go through a lot. But it, if we're going to, is it dominant, is it recessive? If someone were to inherit 2 mutant BRCA alleles, they might not make it to birth. All right, so it can display homozygous lethality. And so if you're asking, here's a gene which, when mutant, causes breast cancer risk, that's actually the intermediate phenotype of heterozygotes, because if you had 2 of these mutations, if they were serious mutations, which say produce no function at all, then no protein at all, then the individual might not make it to birth. And so that being alive with a breast cancer risk is intermediate between being normal and then not making it to birth. So a lot of the disorders which we call incomplete, which we call dominant disorders, are actually incompletely dominant disorders. Now, one of the points before getting into codominance is sometimes we can use the term overdominance. And in overdominance, kind of the implication is that the heterozygotes are actually healthier than the homozygous conditions. Now, unfortunately, it could be used in two different scenarios. So for example, you could have, say, two purebred lines of, say, dog. And you could say, here's a very, inbred, purebred line. And, the purebreds, they sometimes suffer from this disorder, like they have, hip problems. And the purebred line here, they have another disorder. But when they're interbred and you have a mixed dog, not a purebred, more of a mutt, then they tend to be healthier. So you could use the term overdominance in that sense. But that's referring to like all of the genes. All right, so when you ask, why is this mutt, healthier than either two of those purebreds, it's not referring to this specific gene or as a kind of like a sum of all of their genes, not that they're heterozygous for lots of genes. But getting back to sickle cell anemia, we can see an example of overdominance here. So first off, here are human red blood cells, and here is a parasite living inside them. The protist Plasmodium is an intracellular eukaryotic parasite where it then lives inside human cells. And just in the course of my teaching this course, I used to use the statistic that 1 million people die every year of malaria, thanks to great prevention efforts and great healthcare efforts. The last I checked, it's half that, but still, this is one of the leading causes of human mortality. More people die of malaria than the majority of other single causes, certainly diseases. And so resistance to malaria, resistance to malaria would be a great advantage, simply because it is such a great cause of mortality, especially if you're living near the equator where Plasmodium is prevalent. And one of the things that we notice is that sickle cell anemia, if you were to have two mutant alleles, that's a serious health condition, and that might cause you to live a shorter life. But in areas where malaria is present, malaria, once again, kills hundreds of thousands of people per year. And so having two normal beta hemoglobin genes, which would make you more susceptible to malaria, that might shorten your life. But it turns out heterozygotes have, so that sickle cell trait, they actually have some resistance to malaria, and certainly different forms of malaria. Some heterozygotes are highly resistant. And so therefore we would use the term overdominance, because being homozygous for one allele makes you more likely to suffer health consequences of sickle cell anemia. Being homozygous for the other allele makes you more likely to die of malaria. So sickle cell trait, the heterozygotes actually have an increased likelihood of surviving. They're more fit. And so that's what we call overdominance, a case where the heterozygotes are more fit than the two homozygous conditions. And that is why we should always talk about sickle cell anemia, because it's more common than one would expect. When there are mutations which can cause harmful diseases, then generally they're rare in the population. The sickle allele is much more common than one would expect. And the reason is that in some parts of the world, it was an advantage to have one sickle allele. You were more likely to survive if you have one sickle allele. So because there was an advantage to this, and there was an overdominance when it came to being a heterozygote, the sickle cell allele, and thus sickle cell anemia, it's more common than one would otherwise expect. Now, another form of inheritance, which is often confused with this, is what's called codominance. So that's the last one I'd like to mention in this video. And the application, which is most obvious with codominance, is to talk then about blood type. Now, blood type's a little confusing, and when I teach anatomy and physiology, this could be, a good half hour, and I don't mean to do that, but let's just, before talking about the inheritance of it, just, quickly. So red blood cells can express certain glycoproteins, proteins with sugar groups on them, and based on which glycoproteins are expressed on red blood cells, that determines which antibodies our white blood cells can make. So we can make antibodies which would attack A glycoproteins, that would be anti-A antibodies, or antibodies which would attack B glycoproteins, they would be anti-B antibodies. So if red blood cells express A, let's call that a yellow triangle, one wouldn't want to make antibodies which attack A. One doesn't want to make anti-A antibodies if you would attack your own blood. If you have the B glycoproteins, let's say that those are green triangles, you wouldn't want to make antibodies against B if you had B on your blood type, because you would attack yourself. That's an autoimmune disease. And so you wouldn't want to make those. And if you were AB, if you had both the A glycoproteins and the B glycoproteins, then you wouldn't want to make either. So the antibodies which you make in your white blood cells are determined by which glycoproteins then are expressed on your red blood cells. So what's actually being referred to here is there's a protein on red blood cells called the H antigen, to which we then attach sugar groups, making it a glycoprotein. Most, maybe all, of cell membrane proteins are actually not just proteins, they're actually glycoproteins with sugar groups attached. And so sugar groups are attached in a specific order. There is a red fucose, which is attached at this point. And if that's where you stop, then you are blood type O. So individuals with blood type O have this protein with that sugar chain sequence ending with that fucose. That's known as the O glycoprotein. And if your red blood cells only have that, then you're blood type O. However, you didn't have to stop, all right? And so you could then put a yellow sugar here, the N-acetylgalactosamine. That would give you the A glycoprotein, and that would mean that there would be an A in your blood type if you did that. So if you attach this with the enzyme here, that makes you blood type A. Or if you had an alternate form of that enzyme, instead of attaching the yellow sugar and giving you the A glycoprotein, you could attach the green galactose, and then this would then be the B glycoprotein. And so the genes for determining these major classes of blood type's a little more complicated than this, but we should concern ourselves with that right now, is you could either make blood type O, once again you stop with this sequence. If you attached the sugar group, then you were blood type A with that yellow group, blood type B with this group. But because you inherit alleles from both your mother and father, it's possible that your mother told you to do this and your father told you to do this, in which case you would be blood type AB, as I will explain. So as we get to then blood typing, how do we explain its inheritance? Well, I'm going to want to refer to codominance, but also you'll see that I'm referring to something known as multiple alleles. When Mendel first described inheritance, he depicted each gene as having two alleles, like the purple flower allele, the white flower allele, the tall pea plant allele, the short pea plant allele. But as I've just said, even if you just have two in your cells, the population is bigger than you, and there could be more than two. There could be alleles that you don't have. So there are actually a lot of alleles for blood type, but there's three common ones. There's A, B, and O. O is recessive to A. So if you are homozygous for the A allele, let's write that as capital I for immunoglobulin A. So if you're homozygous for this allele and someone is homozygous for the O allele. O is recessive because all of these children will be heterozygotes, but they will then have blood type A. So you can be blood type A with 2 alleles, or you could be blood type A with just 1 allele. And this is a classic example of dominant and recessive inheritance. These heterozygotes are no more or less blood type A than this homozygous individual is. So O is recessive. Now, if we had an individual who was heterozygous for blood type A, then half of his sperm have an A allele, but then half of his sperm have a recessive O allele. That means that half of his children would get an A allele. They would be blood type A if his partner's blood type O. And then half of his children would get a recessive O allele, and they would be blood type O. So the O allele, the lowercase I, is recessive with regard to blood type A. The O allele is also recessive with regard to blood type B. In the same way, if you had an individual who was homozygous for the B allele, all of the children of this couple would get a B allele from their father and O allele from their mother, and they would all be blood type B. If a heterozygote then had children with someone who was homozygous, then you would notice that half the children would get a B allele from their father, half their children would get an O allele from their father. So half the kids would be blood type A or blood type B. So then finally, what would happen if someone who's homozygous for blood type A, I'm sorry, this individual should be homozygous, and their partner homozygous for blood type B, then what happens? Well, now all of the children get an A allele from their father and a B allele from their mother, but neither one is dominant with regard to the other. So you would not be A or B, you would actually be both. You would put A glycoproteins on your cell membrane, and you would put B glycoproteins on your cell membrane. In the previous example, you would make both yellow and green triangles, so you would be both. And that is a form of inheritance known as codominance. The heterozygotes are not intermediate between the two homozygous conditions. They have both of the homozygous conditions, both being blood type A and blood type B. Now, obviously, this could be a little confusing, especially if individuals are heterozygotes, all right? Because if they're heterozygotes, they can actually get a quarter of the children blood type AB, a quarter of the children being blood type A, a quarter of the children being blood type B, and a quarter of the children being blood type O. And so we want to look at Punnett squares on how we would solve this. So once again, in a Punnett square, we know that individuals are diploid and that they have 2 copies of each gene in their cheek cells, their muscle cells, their bone cells. But when they make sex cells, their sex cells have one or the other, but not both. So this woman would put a recessive O allele in her ova. This man will put a dominant A allele in his sperm. And when they fuse, then all of these offspring then are going to get a dominant A allele from their father, a recessive O allele from their mother. And since A is dominant to O, they would put those yellow A glycoproteins on their red blood cells. So they would be blood type A. In the same way, if you had a couple where someone was homozygous for blood type B and the other one homozygous for the recessive allele O, then all of the children would get a B allele from their father in this case, a recessive O allele from their mother, and then all of them would then have instructions to make the B glycoproteins on their red blood cells. All of them would then be blood type B. If, however, one parent was homozygous for blood type B and one parent was homozygous for blood type A, neither of those hides the other condition. So all of the embryos are going to receive a gene from their mother saying to put yellow A glycoproteins on their red blood cell membrane. All of the embryos are going to receive a B allele from their father telling them to put green B glycoproteins on their red blood cell membranes. And so then thus all of them would then be both, in that they would have both yellow and green glycoproteins. So they would be both A and B simultaneously. They would be blood type AB. This is an example of codominance, where the heterozygotes then would not be intermediate between the two homozygous conditions. They would actually have both. And I'll give you a few more examples. So for example, if one were looking at lentils and there was one trait, where the lentils were spotted and another where they were dotted, you could then actually have codominant conditions where the lentils are spotted and dotted. If you were looking at female calico cats, and it has to be female, but we'll get to that later. If one parent had orange spots and one parent had black spots, then you could have the offspring having both. So black spots isn't dominant to orange or vice versa. The calico cats would have both orange and black spots. So that's an example of codominance. Now, once again, this is confusing because there are multiple alleles in the population now. So notice that there's not just A and B, there's also A, B, and O. And so if you had two heterozygotes, one individual heterozygous for blood type B, one individual heterozygous for blood type A, that there would then be 4 different genotypes in the next generation. So you would have some receiving 2 dominant alleles, some with a dominant A and a recessive O, some with a dominant B and a recessive O, and some with 2 recessive Os. And so therefore the blood types in this generation would be AB, A, B, and O. So these are, this is an example of codominance. And once again, this would be common, but to some degree it depends on how you look at it. So some of you may actually have been tested for which alleles of the apolipoprotein E gene that you have. If you did something like 23andMe, you would have gotten data back on which alleles of the apolipoprotein E allele you have. And this is a predictor of, among other things, heart disease and Alzheimer's. There are multiple alleles. Once again, I'll get back to that. So there's an E2 allele, E3 allele, E4 allele, and those are the more common ones. And the reason that they're different alleles is because if you were looking at the proteins are long chains of amino acids. This one at this spot has a cysteine at spot 112. At this spot, it has a cysteine, spot 158. This one at spot 112, it has cysteine, but at 158, it has arginine. This one at spot 112 and 158, there's arginine. So there's different amino acids. And so therefore you can have more than 2 alleles, as we'll get to. So when we look at inheritance, it turns out that when we make cholesterol particles called LDL particles, we'll use the apolipoprotein E's, which we have in our lipoproteins. And if you make more than, if you're a heterozygote, then you will say put both, say E2 and E3 in these lipoproteins. If you are a heterozygote with E3 and E4, you would use both. And it ends up mattering in that if, for example, you are homozygous for E4, then you are at a much more elevated risk of both heart disease and Alzheimer's disease. So for example, I know I have inherited one form of E4. So that means I am slightly, or I have some increased risk of heart disease because I have one E4 allele. Had I received 2 E4 alleles, I would have a high risk of heart disease and Alzheimer's. And E4s, I mean, E2, E3, E4, they're common in the population. And so lots of individuals in the population have an E4 allele for apolipoprotein E. So here's an example of codominance, that you are expressing both. So if you are E2, E3 heterozygote, you put both of those alternate forms of the protein in your lipoproteins. If you're E2, E4, you put both. So that's why this is called codominance. Now, once again, how we classify this could vary. If we wanted to say like being put at a very high risk of heart disease and Alzheimer's, then E4 is recessive. If you're going to include even moderate risk increasing, then E4, because the individuals with one copy of E4 have some risk, not as high as E4, then we could say that's incomplete dominance. Then finally, and I've mentioned this a couple of times already then, this idea of multiple alleles. This would also be something that Mendel did not notice. Mendel simply noticed, once again, tall and short, purple and white. But if a protein can have hundreds of amino acids, or hundreds in a chain, then if the first spot, instead of the amino acid that's supposed to be there, it could then be changed to the amino acid tryptophan, or lysine, or glutamine, or methionine, or cysteine, or whatever. There's 20 different amino acids, one of which is right and potentially 19 are wrong. So at spot 1, you could have a lot of different mutations. You could say, here's a mutant, here's a mutant, but they're different. At spot 1, this person puts this amino acid incorrectly in that spot. A different person could put a different amino acid incorrectly in that spot. And now we go to spot 2, residue 2, same thing. Residue 3, same thing. So there can be lots of mutant alleles, lots of variant alleles. We call something a mutant if it's less than 1% of the population. If more than 1% of the population has a certain allele, even if it was originally a mutant, I mean, it was, as long as it's different from the wild type. But now it's just called an alternate allele. So that some mutant alleles, then become more common in the population and then just become alternate alleles. Then there are other forms of mutation which can be part of a later discussion. So some mutations could actually like truncate a protein, stop it too early. All right. So for example, perhaps the most important protein in the human genome in preventing cancer is called P53. It's important in preventing breast cancers, colon cancers, actually cancers all over the body. A lot of people have cancer because they have a mutant form of P53 that doesn't work as well as it should. All right, so mutations here are serious, and perhaps we would like to screen people. So if you were to ask, all right, well, the P53 protein, it has a lot of amino acids in an order. That's how you make the protein. Let's list them. So let's give them an abbreviation. So there's 20 amino acids, and let's give each amino acid a letter that represents it. So like M would stand for methionine, P would stand for proline, B for valine, etc. And so what you're about to see, this is the sequence of the normal form of P53. Now, what is a mutation? A mutation would be a change, all right? So keep in mind, so some people could have a mutation here at this spot, but some people could have a change at this spot, or this spot, or this spot, or this. So there's a lot of room for mutation. And since a mutation could say stop the protein early, or it could put a different amino acid in place of one that's supposed to be there. If there's 20 possibilities, all right, then we, instead of putting a cysteine, all right, let's just pick this cysteine. Instead of putting cysteine there, we could put a serine there. That would be a mutation. Or threonine would be a mutation. Or glutamine, or aspartic acid, or alanine, or phenylalanine, or histidine, etc. And so Mendel's original model, where each gene only has like 2 alternate forms, the purple or the white, or the tall versus short, we now understand that there's far more to it than that. And so if you were screening people for for mutant alleles, you could then, say, well, at spot 249, which should be this amino acid, is now that amino acid. And that was observed in someone who had liver and cervical cancer. Or you could now see, here's someone who had liver cancer. Here's a different spot of the protein. And this amino acid, which is typically there, was now substituted for that one, etc. And so, once again, the point is that there are multiple alleles in a population. And if you were studying mutants, you wouldn't just say, like, this person has a mutation. Which mutation? Some might be more serious than others. So, once again, just this idea of multiple alleles, if we were looking at the mutations of P53, which have caused cancer, we would unfortunately see, that some patients had a mutation at this spot, replacing this amino acid for that one. Some patients had a mutation at that spot, or at that spot, or at that spot, or at that spot. So there's lots of potential mutations. If one were then to study, a research organism like fruit flies, the same thing would apply. So let's say there's the white gene whose mutations can cause white eyes. Well, it's a big gene. It's got a chain of amino acids. And so therefore, there could be multiple mutations in the same gene. So like if you went into a fly stock center and you wanted to order mutant fruit flies, you might say, oh, look, here's a bunch of different mutations. I can get white, I can get buffy, I can get apricot, I can get all of these different mutations. These would actually be mutations of the same gene. So notice that they all have this letter W. But now we have to say, this is the apricot allele of this. So there is like a problem. Now that there's multiple alleles, how do we represent this? Mendel, I'm sure a lot of us going back to high school use capital and lowercase letters. Well, if there's only two alleles, that's fine. The dominant allele, that's the capital letter. The recessive allele, that's the lowercase letter. But if you're going to be dealing with hundreds, thousands of different alleles, then maybe you want a different way of notating them, like this. This would be the white allele, white gene, but the allele which causes the apricot eyes. All flies have a white gene, but most flies have the wild type. So we often indicate wild type with a plus sign. So if you saw white plus, it just has the normal form of the white gene that gives it the normal brick red eyes. Okay, so point is, Mendel founded genetics in his garden, and before Mendel, genetics was largely based on errors and superstition, and so kudos to Mendel. But obviously, he did not observe everything in his garden. We are still trying to discover, how traits in humans and other organisms are inherited. So there are extensions to Mendelian inheritance, or exceptions to Mendelian inheritance, things that Mendel did not observe, which we have learned subsequently. And these would include things like incomplete dominance, where the heterozygotes are intermediate between the two homozygous conditions. Codominance, where heterozygotes have both homozygous conditions. Overdominance, where heterozygotes actually may be more fit than either of the homozygous conditions. And then multiple alleles, even if individuals only have two forms of a specific gene, that doesn't mean that the population is limited to just two. So these are some of the extensions to Mendel. There are more, which I will cover in the next video.